yesterday we did a lot of single neuron stuff and some time series analysis. Today is going to be about dealing with high-dimensional data sets in neuroscience, focused on neural recording data sets, because that's what I know. We'll talk about projections of neural activity, followed by a bit of dimensionality reduction and classification. After the intro talk for this philosophy discussion section, Sarah Soya is going to be joining us over Zoom. She's a senior theory PI at Northwestern and has spent a lot of time thinking about concepts of neural manifolds and how neural activity lies in a low-dimensional subspace of the possible space. And she'll be going over, I think she has some slides. She'll be going over some concepts from that. So it'll be a little more organized than the philosophy yesterday, but should be fun. Sarah's really awesome. So the reason that I started with this image, was from a book cover, is to introduce the idea of projections in neuroscience, and that different projections of your data can produce different signals. And when you have high-dimensional signals, you can make lots of different readouts from that high-dimensional population to decode different pieces of information. So that you kind of see here, you have a three-dimensional structure, and if you take different two-dimensional projections of that structure, you make different observations. You see an E or a G or a B. Another example of that I found online is, you may have seen this kind of artwork where you have a bunch of like objects hanging in 3D space, and if you find the right perspective to view those objects, you'll see that they assemble into an organized structure like a face. So when you take things that exist in a high-dimensional space, you can project them in different ways. And in some ways, you'll see that there's structure that emerges from those projections. And in other ways, it might just look like a bunch of random things floating in space. And this is interesting in neuroscience, both from a scientific perspective and as a tool that we use to make sense of our neural data. So to start, I just wanted to go through the idea of projections from a neural readout perspective. So say you have two neurons in the brain, neuron 1 and neuron 2, that have firing rates, and you have a third neuron that is receiving input from these two cells. And the input that that third neuron is receiving is scaled by a synaptic weight. So there's a weight on the input from neuron 1 and a weight on the input from neuron 2. Then the response of neuron 3 is going to be this weighted combination of the firing rates of the cells. And in reality, these cells have like threshold nonlinearities and odd things happening. But if you just think of like the total synaptic current that's coming from these two cells, the third cell is seeing a weighting combination of what these two other neurons are doing. And in the brain, this third cell would then be spiking if the input it's getting from the two other cells is above some spike threshold. So this is basically the leaky integrate and fire cells that we looked at yesterday. So for simplicity, we're just going to assume that these weights have some uniform norm, and all we need to do with them is choose how much weight we assign to neuron 1 versus neuron 2. So in terms of what this means for representations of neural activity, if we take this plot of the two firing rates of the cells, we can define the spiking threshold of the neuron. So the cell is at its spiking threshold whenever the weighted sum of the first neuron's firing rate and the second neuron's firing rate is equal to the threshold B. That's like right at the decision boundary of, if you're a little bit above that, you're going to spike, and if you're a little bit below it, you're not. So if we take this equation, we can reorganize it to be in terms of a relationship between the firing rate of the first cell and the firing rate of the second cell. And now that we have this equation, it's of the form y equals mx plus b. We can plot it as a line in this firing rate space. So this is what you could think of as the neuron's decision boundary. If you're at a point that's above this line, if mx plus b is greater, or if the weighted sum of the neural firing rates is above b, the cell is going to spike. And if the weighted sum of the firing rates is below b, the cell isn't going to spike. So if you take some arbitrary stimulus and you present it to these two cells and you see the responses of those two cells, you can plot them against each other and get this point in neural activity space, which we're going to call A. So that corresponds to a firing rate of neuron 1 and a firing rate of neuron 2. What's interesting about this idea of projections is that there are changes in the location of A that are meaningful for our readout neuron, and there's changes that are not. So if you move A up or down in this direction, kind of parallel to the decision boundary, the total input to our readout neuron is not going to change. It's still going to respond the same way if you have neuron 2 super active and neuron 1 silent, or if you have both neurons firing at some intermediate rate. Conversely, if you move A in a direction that's orthogonal to this decision boundary, now you're changing how much or whether or not this postsynaptic cell is firing. So any movement that goes further away from the decision boundary is going to increase the response of the readout cell, and anything that moves you towards the boundary or past it is going to decrease the firing rate. So rather than thinking of neurons 1 and 2 as just two firing rates that are happening in the brain, you can think of these neurons as having kind of a potent mode that changes the readout and a null mode that doesn't change the readout. And this is really what projections are doing, is they're taking different weighted combinations of neurons. And when you do that reduction of dimensionality from 2D down to this 1D value of, to this 1D distance from the decision boundary, you can transform your data in interesting ways. So in math, the concept of projection is pretty much what I just showed you for spiking neurons. You define a decision boundary, a dimension that you're going to project that signal onto. You can compute the projection of neural activity into one direction, based on the weighted combination of the cell firing rates, and you can project it in, or you can define the location of this point A instead of this firing rate of neuron 1 and firing rate of neuron 2. You can define it in terms of the potent dimension, the weighted sum of the cell's firing rates, and the null dimension, which is the orthogonal dimension to this potent thing. So to find that orthogonal dimension, you just find a vector that is perpendicular to the vector defined by these synaptic weights here. So the projection of this response of the cells onto the decision boundary of the cell of our readout neuron, you would define as just the dot product between this set of synaptic weights and this set of firing rates. And it gives you sort of the total input of the two presynaptic cells onto our postsynaptic cell. So it's often more meaningful to think of neural signals in terms of these projections onto particular dimensions of the data instead of just the absolute magnitude of the signal, the straight line from the origin to the point A. This is just to show that you can also compute this projection, this potent dimension and this null dimension in terms of the magnitude of this vector, the magnitude of the firing rates, times the sine or the cosine of the angle between the line to this point and the line that forms the neuron's sort of integration dimension. And you can see that the smaller this angle becomes, the closer this point moves down to being on this potent signal, the stronger you're going to drive this potent dimension, and the smaller the change in the null dimension is going to be if the signal, or the smaller the component of the null dimension is going to be. So projections, as I said, are a really powerful concept in neuroscience because you can have a neural population that has a whole mix of different firing rates, and some readouts of that population could be in a potent space and some could be in a null space. Or if you have a readout, you can have signals that are in the potent space of that readout, and you can have signals that are in the null space of that readout. So where we've seen this applied the most in neuroscience is in motor control, which is if we have an action that we're going to be performing, how do we plan to carry out that action without actually engaging our muscles? So if you think of a monkey who has his finger on a screen and he's reaching from a starting position to a target, there's a series of transformations that have to happen from his eyes and his brain out to the muscles in his limb. So the muscles are receiving input from the spinal cord. The spinal cord is receiving input from some primary motor cortex that's sending a motor command. And, well, that's interesting. But the primary motor cortex is receiving input from higher brain areas that are planning the movement that the animal is going to make. So there's this series of transformations that takes place in generating the reaching movement. And during the course of preparing a reaching movement, we can look at the activity in each of these areas. And what we see is really interesting. During the movement itself, all of these areas are very active. The muscles are engaged. They're being driven by spinal populations that are being very engaged. The spinal populations are driven by motor cortex and premotor cortex, and there's a lot of activity in all of these areas. So that's kind of what you'd expect. The brain is generating motor output, so neurons are spiking. What's interesting is during the planning period, you still see really high activity in brain areas that are involved in kind of motor planning and charting out the trajectory of the reach. But as you go from those pre-motor planning areas down to the muscles, the magnitude of signals diminishes very quickly down to nothing. So you have a lot of activity in your planning region, but that activity isn't being translated to firing of motor pools in the spinal cord, and it isn't being translated to muscle contractions. So somehow you can have relatively the same amount of activity during planning and execution of movements, but in one circumstance, that high motor activity is driving muscle contraction, and in another circumstance, you have just as much activity in cortex, but it's not causing any contraction of the muscles. So there are lots of ways that you could pull this off. Maybe there's some sort of thresholding, maybe there's some sort of inhibitory gating. But if you look at activity in premotor neurons leading to the onset of movement, firing rates during the preparatory period aren't really any lower than firing rates during the movement. So you couldn't just like threshold out motor activity. So there was this question in motor control neuroscience for a long time of how you could have the same amount of activity producing different motor readouts. So a clue to what's happening here came from the work of Mark Churchland in Krishna Shenoy's lab, and he's followed up on this in his own lab at Columbia. They went in and they looked at the activity of individual neurons during periods when the animal is preparing to generate a reach and when the animal is actually reaching. And these plots here are color-coded by the direction of the reach. The animal's just kind of reaching to different points located in a circle around where he's starting. And you see that the activity patterns aren't just kind of a fixed signal that turns on and stays on, but there's lots of fluctuations that are evolving over time. And the activity of neurons prior to reach initiation is often different from the activity of that same neuron. Hello, David. The activity of that same neuron during the reach. So if you go neuron by neuron, not only is there this confusing question of why some activity is driving muscle contraction and other activity isn't, but also it's pretty difficult to interpret what these individual cells are representing. If they're tuned for a reach direction or a pattern of muscle contraction, they're kind of all over the place. And what Mark Churchland and the Shenoy lab showed was that if you take neural activity and you project it down into a low-dimensional space and you do the right transformation of the data, what you see is that motor activity kind of lies in two orthogonal dimensions. So what I'm showing you here is data from two animals. Each gray point here is when the go cue appeared and the animal initiated a movement. The lines in blue are the activity during the preparation period, and the lines in green are the activity during the movement period. And after staring at these trajectories for a while, what they realized is that these are two orthogonal signals. When the monkey is preparing to reach, there's a movement of the trajectory in one direction or another along this axis. And then when the reach is taking place, there's movement that's largely orthogonal to that along this second axis here. And I should say these two clouds of points are just corresponding to trials when the animal's reaching to the left and trials when the animal's reaching to the right. So it's really the same thing that we saw in this idea of projections of neural activity and spiking thresholds. If you have two neurons in the brain that are, just for simplicity, directing straight to muscle, you have potent dimensions and null dimensions. You have weighted combinations of those neurons where the weighted sum is going to change as their firing rates change, and you have weighted combinations of those firing rates where the sum of them is not going to change. And basically what they showed in a series of follow-up studies was that during the preparatory period, neural activity is moving in such a way that the total input to the muscle isn't changing. You're increasing the firing rate of one neuron and decreasing the firing rate of another, but you're moving in this null dimension. So the total input to the cell, which is projected along this green line, isn't changing, or to the muscle. So they likened this to kind of storing up potential energy in a system. You're taking a cart and you're pushing it up to the top of a hill. you are changing the firing rates of neurons. You're perturbing the system from its baseline state, but you're doing it in a way that isn't changing the output of motor cortex to muscle. Then when the actual reach period is initiated, the position that you move during the preparatory period determines how the activity in your system evolves over time. And once the go cue appears, neural activity will circle in this orthogonal dimension. And now you're getting a signal that is going in the dimension that is driving increases or decreases in muscle activation. So you push yourself out here, and then once the go cue appears, you change your pattern of activation and contract the muscle or relax the muscle. And so if we take readouts of the neural activity on this output null projection, you see that there's an increase in activity that appears when the target appears. It remains high up until the go cue appears, and then you get these fluctuations as it kind of follows this trajectory back to baseline. And in the output potent dimension, the dimension that changes the level of recruitment of the muscle, even though neural firing rates are changing, there's no change in the total input to the muscle until the go cue appears and you start to see a change in the summed firing rate of the neurons that drives the contraction of the muscle. So the firing rates of the individual cells are going to look like these crazy wavy messes of signals, but if you take the right weighted combination of neurons, you can find this output potent space and this output null space, one of which drives motor contraction, the other which doesn't. So the important thing to take away from this is that it's only the activity in the potent dimension that is affecting the contraction of the muscles. And we have a mechanism by which you can change what a population of neurons is doing. You can move its state from A to B without that change being detected by areas that these neurons are projecting to. So long as there are more neurons in the brain than there are muscles in the body or downstream targets of these neurons, there are always going to be output null dimensions. If you define a projection to a muscle as a set of synaptic weights from neurons onto motor neurons, only activity along that dimension is going to change the activation of motor neurons, and any change in activity that's orthogonal to that is not going to have any input or any effect on muscle contraction. So this is it takes a while to kind of wrap your head around it, but I think this is a very powerful concept in neuroscience, that you can have lots of changes that happen in neural activity that aren't detected by downstream areas. And it's only when you have the right pattern of activity that you're able to drive activation of a downstream brain area. Amit?
Yeah, I'm a bit confused. So premotor cortex does not project the muscle.
Right, yeah, this is a cartoon version. Premotor projects to motor cortex, so you could have preparatory activity in premotor that doesn't drive motor cortex activation, or drives it to a smaller extent.
Okay. And second, this is a nice way of decoding information.
Mm-hmm.
How do we know it's actually happening in the brain? Motor cortex actually, this is the way it plays out.
Yeah, that is a separate thing. Mark Churchland is very much trying to make the argument that this is how motor cortex works, and furthermore, that where you get pushed by your preparatory activity determines how your reaching activity evolves. And he argues that motor cortex is a dynamical system, so how you set your initial conditions determines how it's going to evolve from there. Whether that is actually the case for motor cortex and other regions, I think is still a subject of some debate. This idea of neural populations as dynamical systems works for some regions, probably isn't a good description of other regions. So yeah, this as a concept is something that's definitely possible, definitely is happening in brain areas. You can have changes in neural activity that are not detected by downstream regions, but this whole narrative of how motor control happens, I think the Churchland lab has made a good case for it. A reasonably good case in the Kaufman lab, but yeah, I think the total jury is still a little bit out.
But theoretically, you can extract features of this dimension that you can maybe test in an experiment, whether these features are important.
Yeah. Yeah, they've done a lot of follow-up experiments trying to argue that motor cortex acts like a dynamical system. they had this paper on like tangling of neural trajectories. So if the evolution of neural activity is just shaped by the weight matrix, then 2 points that start out close together aren't going to flow in opposite directions. They're kind of driven by the same dynamics. So they've done some work to try to make the case that motor cortex is better thought of as a dynamical system. Yeah, I'm not going to get into that today. This is more just to describe this idea of potent and null dimensions. But there's, maybe Sarah will talk about it. We'll see. There's a whole discussion about, yeah, how to think about motor cortex to be had. Hope that was reasonable. Any other questions? Oh, Mingyu? Oh, sorry, you're muted. 1 sec. Or I had you muted. You can talk now.
Okay, so the null projection is in terms of the as an instruction for the muscle projection, right?
Mm-hmm.
It's not probably this pattern is not captured by this muscle downstream, but it has some other meaning for the other pathways. Probably serves as other function. Is that?
Yeah. Yeah, if you had a different weighted combination of neuron firing rates, you could find just the preparatory activity and not the movement activity. So maybe, yeah, so you can use this high dimensional representation to encode multiple signals within the same neural population. I guess an example would be our imaging in VMHDM. We see a dimension of the neural activity that's kind of like the intensity of the stimulus versus a dimension that is the identity of the stimulus. And they're two independent signals. So you can have multiple readouts from the same set of cells that do different things.
So this null dimension is just something that people haven't been recognized the function.
No, I'd just say that in this particular example, the function of the null dimension is to allow motor cortex to change its activity and set itself up to generate a reach. So the thinking is you couldn't produce the firing rate pattern you need to activate muscles from baseline. You kind of have to start by priming yourself, moving your activity into a certain space. And then once you initiate the reach, the activity drives this change in firing rates that activates the muscles. Yeah. Okay. So that is the concept of projections. And now for the rest of this intro talk, remaining half hour or so, we're going to talk about forms of projections of neural activity. The first is dimensionality reduction, and the second is classification. And both of these are just different strategies for taking projections of neural activity. So I think you guys probably learned principal component analysis in the day one tutorial. So I'm not going to do the full derivation, but I wanted to briefly recap it, which is if we have a population of neurons, or if we even have just two neurons, we can plot the firing rate of neurons as a function of time or as a function of stimulus number against each other. And we can see that there's some covariance between these cells. On some trials, they're both firing a little bit more. On other trials, they're firing a little bit less. We can use projections to create different summaries of what these two neurons are doing. So the strategy that you're using in principal component analysis is you're finding a projection from 2 dimensions down to 1 or to a couple that preserves as much information, as much variance as possible. So when you're taking a projection of neural activity, you're again just taking a weighted sum of neural firing rates. So each of these individual trials is going to get projected down to this 1D summary. So rather than encoding this 2D signal of what neuron 1 and neuron 2 are doing, you're going to encode this 1D signal of how far you are along this potent dimension. And you're throwing out any information that's in this null dimension. And again, this dimension is just defined by taking a weighted combination of neural firing rates. Like that. You can take different projections of neural activity and produce different one-dimensional summaries of the data. So this is something that we kind of saw before. By rotating the line, by changing the weights on neuron 1 and neuron 2, you can produce projections that capture different readouts of what's happening in the neural population. This is kind of like the blocks with the shadows I showed you at the start. We're getting a different 1D portrait of this 2D signal. And when you're performing principal component analysis, you're just picking very specifically the dimension that maximizes the variance of the projected data. So you're picking your potent dimension, your S, such that you're maximizing the variance of points along this dimension. So we can define the variance in terms of the distance of each projected point from the mean of the projected points, and get this equation for the variance of the data set. And this is just the term that we want to maximize by picking our weights. And if you go through the full derivation, you'll see that the set of weights that maximize this term, subject to a constraint on their magnitude, is the first eigenvalue of, or eigenvector of your covariance matrix of your data. An interesting facet of PCA is that the dimension that's maximizing the variance is also the dimension that minimizes the reconstruction error of your data. So the reconstruction error is how much you've thrown out from the data set, and it's basically the magnitude of these red lines here. And by picking a dimension that maximizes captured variance, you're also minimizing the information that's lost. Yeah.
Do dimensions have to be orthogonal from each other?
General, the components, yeah, by construction, they're orthogonal to each other.
I mean, could you have a PCA where you specify a rotation that's not orthogonal, and is there ever any use of that?
Yeah, so there is a variant called independent component analysis that doesn't impose orthogonality, but it imposes a constraint of statistical independence. Orthogonal signals are nice because you know that they are independent of each other. An increase in one dimension is not going to change where you are on the second dimension. But yeah, it's...
well, it's just possible that you could find components that do vary with each other, but...
Yeah. Or you could have signals that covary with each other, but that get projected onto multiple PCs. Yeah. So one of the common complaints about principal component analysis is that it's unsupervised. We're not giving it any information about the identity of the stimulus or any structure of our experiment. We're just kind of finding this single dimension that captures as much as possible. So if we have, say, 3 different stimuli that we're presenting in red, green, and blue here, and we project their signals onto the first principal component, we might get a representation that's preserving as much variance as possible. It's capturing this difference in the sum of the firing rates and this higher activation of the neurons by one stimulus versus the others. But it's not really doing an excellent job of separating out the representations of these different stimuli. It's kind of pushing the green and the blue stimulus on top of each other, even if their original representations are distinct. And this axis, because it's just trying to capture variance, and you have this one signal that's way out here, is kind of getting biased by these points here, and is losing any structure that distinguishes between these two conditions. And if you define your second principal axis as something that's orthogonal to it, you just kind of get this mixture of signals. And neither of these two axes is really ideal for capturing information about stimulus identity. It's just kind of gotten mixed when we find this rotation. Okay. I wanted to toss in a quick aside about concepts of dimensionality reduction as they relate to neuroscience. There are kind of 2 schools of thought about the utility of dimensionality reduction for neuro. The first is just that it's a way of making sense of your data. You have 200 neurons that you've recorded. You don't want to have to stare at all of them, so you look at their first couple principal components instead and you interpret them. That's what you could think of as the weak principle of dimensionality reduction. It's a useful tool for thinking about what your neurons are doing. The other way that you see dimensionality reduction methods used in neuroscience is this strong principle that neural activity does inherently lie on a low-dimensional subspace of the full-dimensional space. So the weak principle says, as you add in more and more neurons, your dimensionality is going to keep increasing because there's more and more signal that you have to explain. In the strong principle, there's some underlying inherent dimensionality of the neural activity. And if you record 1,000 neurons versus 10,000 neurons, the dimensionality might not change so much because each of our neurons isn't really an independent agent that's doing its own thing, regardless of what all the other neurons are doing, but neurons are acting as a population to drive speech and movement and motor control. And so there's this upper limit on the dimensionality of a neural population that comes not from the number of neurons that you record, but from the underlying tasks that those neurons are performing. Right, so that is PCA and this idea of projecting neural activity. Useful for summarizing things, might not tell us much about underlying structure of our experiment. Just to provide another perspective on PCA before we talk about other projections of your data, I wanted to present the same thing from a different framework. So rather than thinking of PCA as just finding an axis on which to project your neural activity, you can think of dimensionality reduction as constructing a hypothesis about how the data was generated. So if you think that the observed spikes are noisy observations of some underlying signal, then that is a hypothesis that you could design a form of dimensionality reduction to test. You could say, well, I think that my neural activity is being driven by some low-dimensional pattern of population dynamics, some low-dimensional task that the neurons are performing. So if we take this perspective on things, we can start to think about neural activity in the context of this representation of latent variables. And we can think about low-dimensional summaries of neural activity, not as a way of just describing what our data is doing, but as a way of finding the actual underlying signals that the brain is encoding. So we talked about encoding yesterday in this idea of Bayesian inference and relating converting stimulus feature representations into spiking of neurons. And in the framework that we discussed yesterday, you had some known observation of spikes, and you had some model parameters that you fit, and you had some variable X, like a stimulus feature, that you were encoding into the firing rates of cells. So your spikes are known and your stimulus features were known, and you were fitting some model parameters. And maybe you had some Poisson observation model for how the underlying spike rate was being translated to the firing rates of individual cells. And then what we did yesterday was find the most likely value of the parameters that translated our stimulus into our spiking of neurons. When we're doing PCA, we're essentially doing the same thing. It's just that we've replaced our underlying spike rate with a set of latent variables that are driving the observed spiking. So you still have some parameters by which an underlying signal is giving rise to an observed set of spikes from a bunch of neurons. But in the case of dimensionality reduction, rather than trying to relate your observed spiking to the climbing speed of a mouse or the intensity of a pixel in an image that's being presented to the brain, you're trying to relate neurons to some low-dimensional thing that the brain is encoding. And when you're doing PCA or when you're doing any form of dimensionality reduction, what you're actually doing is you're trying to find these latent variables that are being encoded by the population. So PCA is one strategy for doing this, but there's a whole family of strategies that make different assumptions about what these latent variables do, what their structure is, and how they are being represented in the firing rates of neurons. So computationally, this seems like a tricky problem. You now have one set of things that are known, but you have unknown model parameters and you have unknown latent variables. So you can ask, well, how am I supposed to find these things? And in addition to just PCA, where we have a concrete analytical solution for our latent variables, you can fit any form of latent variable to your neural data using expectation maximization algorithms. So you make some initial guess for what your model parameters are, you impose some structure on what your latent variables are doing, and you can iteratively fit a model that best relates your predicted neural activity to the latent variable that you've defined. So EM is something that you'll see often in stats and optimization, and it's just kind of this iterative process. You make a random guess for your parameters, you use those guessed parameters to compute the expected value of your underlying latent variables. You then re-estimate your parameters in terms of your estimate of latent variables, and you iterate this until you converge on a solution. So rather than just like analytically solving for your latent variables, you're doing this wandering convergence on an estimate of these signals. So if we have observed spiking activity of a neural population, and you have some assumption of how your observed activity relates to your latent variables, you can create different summaries of what your neural population is doing. And different forms of summaries have different meanings and different names that have been given to them over the years. So one that you could do is you could say each of my latent variables is just going to be random, Gaussianly distributed, has zero mean, and has uniform variance, and there's no correlation between neighboring time points. And you could say, well, all of my observed neural firing rates are just going to be Gaussianly distributed around a weighted combination of those latent variables with some noise in their observation of them. If we take this and think of the same thing in matrix notation, then we're saying that our latent variables are all drawn from this uniform Gaussian distribution, and our observed spiking is a set of weights times those latent variables with independent noise in each neuron. And these I's here are the identity matrix. This W here is now a loading matrix that converts latent variables to observed spiking. And what we've arrived at here is a probabilistic derivation of principal component analysis. We've said that our observed spiking activity is just a set of latent variables times a set of weights. And you can show that if you impose this specific structure, this uniform noise assumption about how latent variables convert to neural activity, you can show analytically that you arrive at the same solution for the principal components of the system. This is one way of reducing dimensionality, but once we've established this framework, we can go back into this matrix notation here, and we can make different assumptions about the system that give us different low-dimensional summaries of the data, different sets of latent variables. So in probabilistic PCA, we have an assumption that each neuron is a weighted sum of principal components with some residual noise, and that noise is the same for all neurons. Factor analysis is very similar to PCA, but it assumes that different neurons have different amounts of noise, that some are very nicely predicted by your latent variables, and some are very noisy observations of those latent variables. So this residual variance matrix, this noise that's describing, or this noise that is contributing to neural activity on top of the latent variables, is independent for each cell. Yeah?
So just to clarify, the results of the probabilistic PCA with the latent variables would only look like the unsupervised version if the latent variables are indeed causing the separation, or not necessarily?
I'm sorry?
So I'm just wondering, in what cases would the probabilistic PCA not look like the unsupervised version?
They should always converge to the same solution.
I see.
Yeah.
Even if the latent variables aren't
Orthogonal?
or if they're not generating the components.
If they're not
causing a set of neurons to change their firing or something.
Yeah. Well, here the latent variables are just something that we're fitting to the firing rates. Like these.
Oh, I see. They're not latent variables that you decide.
Yeah. Oh, yeah. Sorry. Yeah. So the latent variables you find via this EM approach, and yeah, there's no additional information you're providing. You're not giving it like, here's a stimulus or something. You're just finding the signals that best capture the observed spiking activity. And that ends up just being the principal components of the system. Sorry, that was a lot. Are there any other questions at this point? I was kind of rushing to get through this, but yeah?
So I guess high level,
Mm-hmm.
we're doing like it's not like gradient descent, but over like a dimensional. I guess I'm a little bit confused, like how we're descending along like the weight variables. I thought the weight variables are just like a subset of all the variables possible. And so we're just like checking the different combination of those weight variables to see what that's the most functionality.
Yeah, we've come up with a rule for how the latent variables get mapped to the neural activity, and we're just finding the best.
Would it be like exogenous of the model? Is that like how we...
Oh, the model is the rule, right? Like, so you have observed spiking, and you have a set of weights on latents that are predicting that spiking.
Okay.
Yeah.
Can you go back to DTM algorithm, please?
Mm-hmm.
Maybe I didn't understand. Here Z is also a random variable, right?
Yeah.
Okay. So you introduce it iteratively in each step, but then again, in a subsequent slide, you say that it's normally distributed with mean 0 and unit variance.
Right. So when we're initializing, or we are.
So here you say computing the expected value, but then you assume that the expected value is 0.
We're just not placing any constraint on Z. Like we're not saying that it has to be smooth or that it has to have, we're just saying that we a priori, without knowing anything about its structure, are assuming that each time point is independent. And yeah, it's kind of like the
So you're using
initialization.
the standard normal. Okay.
Mm-hmm.
Maybe I need to see it on zero. Okay.
Yeah.
Thank you.
Are there other questions? I know it's a, yeah. Yeah. Yep.
I have a question. When you say, if you go back to slide, the predicted spike distribution, that seems, if I understand correctly, that's not what you're directly trying to predict. What you're trying or generate here, you're trying to generate a dimensionally reduced picture of what a population of what could be thousands of neurons
Mm-hmm.
that are spiking at different rates at any given time are doing. And so it's going to be, say, you have 3 different latent variables here. It's going to be a time varying trajectory through that latent variable space where the point at each time is the weighted combination of those 3 latent variables. But the point in that latent variable space does not directly itself contain the predicted distribution of all of the spikes in all of the neurons that you originally measured. You have to go backwards from that reduced dimensionality value, don't you, to predict what the spike distribution would look like if you unpacked it and went back to the original high dimensional data, like you can do. Adi showed how you can do that with PCA, and then compare neuron by neuron the predicted spike distribution from the actual spike distribution. Is that what that means in this equation? Because what's confusing is you run these dimensionality reductions, you get these plots in this reduced dimensionality space, and that's what you stare at and try to make sense of. And the predicted spike distributions are embedded in that, but they're not explicitly represented in that reduced dimensionality space. Is that correct or no?
Right. So this predicted spike distributions, what you're basically saying is the spiking that we observe from a given neuron is a weighted combination of, say, 3 latent variables, and everything else that we see that doesn't match that prediction of the latent variables is just Gaussian noise on top of that. So this is kind of the assumption that you make in PCAs, that you have some signal in the, some underlying signal that is driving neural activity, and everything else that you see that isn't captured by that signal is noise that's on top of it. So your predicted neural activity from your first couple principal components would just be the latent variables times these weights. You can get back out your like expected values of what individual cells are doing. And the difference between those expected values and the observed values is assumed by PCA to just be observation noise.
Right, but what the output of the dimensionality reduction, the graphical output, whatever you want to call it, the embedding that you present is the reduced dimensionality representation, where if it's from a timing series and you break it up into a series of points, each point represents a compressed dimensionally reduced picture of what the entire ensemble of thousands of neurons is doing in terms of a weighted sum of these 3 basis functions.
Yeah. Right. So there's kind of, you can map from observed spiking to latent variables, and you can map from latent variables back up to observed spiking. And so what I'm showing is that mapping of latents back up to the observed, but you can also take the inverse of that mapping and go from spiking to the latent variables.
Okay, I guess it's the latter that I'm more familiar with.
Yeah.
Talking about taking high-dimensional spiking or calcium imaging data and then reducing it into the slower dimensional space to look at its principal components or main latent findings.
Yeah, but the two are equivalent. If you say your observed spiking y is equal to, well, let's go the more familiar direction. Say you have observed spiking, you multiply it by a set of weights and you get a set of principal components. You just invert those weights and you can go from the principal components back up to the prediction of the observed spiking. Okay. Minus the yeah, minus whatever you're not capturing when you cut it down to a small set of latent variables, and here you're just assuming that that is some noise that's independent. Yeah, I hope that helped. Yeah, so yes, principal component analysis, you're assuming every neuron has the same observation noise of your latent variables. Factor neural analysis, different neurons have different amounts of observation noise. Other frameworks that you could use to try to summarize what your neurons are doing, you could say that your latent variables are a linear dynamical system. So instead of each time point being a weighted, each point in PC space being a weighted sum of neurons, you could say that there's some system where your latent variables are evolving over time, and your neurons are an observation of those latent variables. So this is what RSLDS uses, at the heart of it are these linear dynamical systems. Another method that people have used in neuroscience is called Gaussian process factor analysis, which doesn't assume that your latent variables are a linear dynamical system, but rather it assumes that there's some smoothness of the latent variables over time. So you have a covariance matrix between time points of your latent variables that is has this kernel to it, so it has a degree of Gaussian smoothness to it. So here you're doing factor analysis the way you are up here, but you're constraining your latent variables to try to be smooth signals. This is a method that was developed by Byron Yu for analysis of spiking data. But you can take this in other directions as well. You could say that my observed spiking is being generated by some set of discrete states, and the state that I'm in determines the firing rates of neurons, like what you do in a hidden Markov model. If you're doing independent component analysis, your observation model of your latent variables is the same as PCA, but you are constraining your latent variables to be maximally non-Gaussian. And in a switching linear dynamical system, you have a dynamical system that is constrained on your latent variables, and you have a switching component so that you can sample from different dynamical systems at different points in time. So starting from this basic framework of having a low-dimensional set of variables that are driving your neural activity, you can come up with lots of bells and whistles based on what structure you think is present in your data and how you want to constrain the signal that you pull out of your data. And there's a really nice review by Sam Roweis, Readout, where they present all of these different kinds of observation models within the same framework. So I can warmly recommend this if you want more background on this material. So that is flavors of dimensionality reduction. All of them are just trying to explain neurons in terms of these underlying signals with different assumptions about the signals. But all of the ones that I've shown you so far don't still know anything about the stimulus that's being used to drive the neural activity. So for PCA or for other representations, you're just finding these latent dimensions that are best capturing the observed spread of neural activity. Another very different way of thinking about things is to just focus on capturing some known aspect of the signal that's driving the neural activity, and that's what you're trying to do when you do classification. So like PCA, classification is, at its heart, You can think of it as a way of projecting neural activity down onto a dimension, but you're picking that dimension in a way that's going to let you distinguish between different experimenter-defined conditions, like the identity of the stimulus. And when you find your dimension to project your neural activity onto, you're doing it in such a way that you can maximally distinguish these different stimuli. An example that I'll go through for that is Fisher's linear discriminant analysis in a little bit of depth, but there's lots of different ways to fit classifiers that will give you different rules for drawing this line. So there's kind of two sides of dimensionality reduction. There's the unsupervised approach, where you're just trying to find a summary for what your neurons are doing. And there's a supervised approach where you're projecting your neural activity in a way that preserves a piece of information. And the big tension between these two is sort of, what is the important signal to preserve in the activity of a neural population? Is it the bulk signal, like the thing that's most present in neural firing rates, is the most important thing that stimulus 3 drives these neurons way more than 1 and 2? Or is the important piece of information the identity of the stimulus? Oftentimes, you can find projections of neural activity that preserve relevant signals like stimulus identity, but if you look at the total variance of the data that's explained, if you look at the amount of information that's lost when you project down to this particular dimension, you'll find that, yes, you can find these signals, but it captures a very small fraction of variance. You might only preserve 10% of the total variance of these 2D points when you project it down to this low 1D space. Versus if you did PCA, you might preserve, say, 50 or 60% of the signal. So there's this kind of this tension between preserving variance and pulling out meaningful signals. And it's not really clear what projections in the brain are doing. Are they pulling out the signal that is the dominant activity of the neural population? Or is the dominant activity just kind of some artifact of computation or connectivity? And the actual meaningful signal is some weighted combination of neurons that preserves some interesting information about the stimulus. So just to go quickly through linear discriminant analysis as a way of decoding out information, this is just one way of drawing a decision boundary between two conditions that makes a particular assumption about the two conditions that you're trying to separate. So again, we're going to set a decision boundary, which is based on a weighted combination of the firing rates of these two neurons. And now we're going to classify whether the stimulus, whether the stimulus was stimulus 1 or stimulus 2 based on whether you're above or below this decision boundary. And this is what's called a linear classifier because you're just taking, again, a linear projection of your signal onto this boundary. People like to use linear classifiers in neuroscience because they look kind of like neurons. If you can decode out a piece of information from a neural population using a linear classifier, then there could ostensibly be a neuron somewhere in the brain that's taking that same linear combination of firing rates and is decoding out that piece of information itself. So for Fisher's linear discriminant analysis, the rule that you're following to find this projection is just this simple goal of maximizing what we call the separability of the two conditions. So you take the two classes of data. Now we're going to use our knowledge of the underlying stimulus that drove the firing rates of the cells. We're going to try to draw a line, a weighted combination of neural firing rates, such that when we project the means of the two stimuli onto this line, we're maximizing the separation between these two means. And we're normalizing that by scaling that separation by the variance within each of the projected classes. So once we take all of these points and we move them down to this line, what is the spread of points along this line? And that is just defined by this, the variance is the firing rates of the cells projected onto the line minus their mean. So you can solve for what that is as this term here. So this gives us an equation for the separability of the cells. And from the separability, we can solve for the value of W that maximizes this signal. And just get that in terms of the variances of the two projected distributions and the difference between the means of those distributions. So this gives us a set of weights that defines our projection dimension. And then our decision boundary we usually take as just the midpoint of these two distributions. So you take the sum of the mean of stimulus 1 distribution, the mean of stimulus 2 distribution, average them, and project it onto the same dimension. Yeah? Pretty much last slide. So, right, we can find a set of weights that maximizes separability. I luckily tossed out all the slides on other linear classification methods, but there are, the same way that there's many flavors of dimensionality reduction, there's many flavors of classification that are basically different rules for drawing this axis. And in general, I think it's worth thinking in neuroscience what we want to find, what trade-off we want to strike between preserving neural variance and preserving experimenter-defined information about the stimulus. So yeah, I will leave us off there and let you guys get into coffee break. After lunch, we're going to go through just a handful of methods for making sense of high-dimensional data and get an intuition for what they do. This is mostly based around data I worked with in the Anderson lab. So you have neural recording data, you have behavior annotations, and you're trying to say something about what is happening in this big data set. So we'll go through a couple approaches to making sense of neural population data based on cell tuning for behaviors, some decoding, some measures of separation, and just general dimensionality reduction methods. So yeah, I'll take questions, or maybe we can go to coffee and we can talk there. Yeah?
Can I say the PCA is mostly starting from the neuronal activity point of view and trying to have the implicit assumption that the most variance might be the most interesting dimension for the neuron, whereas linear discriminant analysis is starting from the stimuli and thinking that the different stimuli and how we classify them would be the most interesting dimension to go from. So it's just like different approach from different two.
Exactly, yeah.
Is there a metric by which you get a sense of how separable, for example, an experimental stimulus are from each other?
Yeah, I think we'll go over that after the break. Different ways of measuring how dissimilar two things are from each other. Or after lunch. Yeah. Oh, there's a question in the chat. The next part of the lecture, so in 12 minutes, Sarah Soya is going, sorry, this is David in chat. In 12 minutes, Sarah Soya is going to come and talk to us about manifolds in neural activity, and then the practical session is after lunch, which is at 1 p.m. Yeah, so either 10, 12 minutes or at 1.
But this section here that you are describing on the slide.
That's at 1 p.m.
That's at 1.
Yeah. Okay. Let's grab coffee. All right. Yes? Oh. Okay.